Hello everyone. Today, I'd like to share my experience using a set of tools to create a presentation video for a paper published in Nature, along with some of the challenges I encountered along the way. This is the front page of the paper, which is in a research area I'm quite familiar with. To make the content more accessible to viewers outside the field, I designed the following introductory section to help set the stage. The central focus of this study is the process of transcription initiation. Although the term transcription may sound technical, it can be understood as the very first step through which DNA carries out its biological functions. The importance of DNA is widely recognized. At the macroscopic level, it is reflected in the shared physical traits between parents and their children. At the microscopic level, it ensures the proper functioning of cellular activities. For these reasons, understanding how transcription begins is fundamentally important. Next, we will use a concrete animation to visually illustrate the transcription process, completing the introduction to our topic. Next, we move on to an interpretation of the study. The overall experimental workflow involved in vitro purification of transcription-related protein components, assembly of the complexes, and activation of transcription, followed by sample preparation after the reaction was halted. Cryo-electron microscopy data were then collected and used to determine high-resolution structures. During the early stages of protein purification, a wide range of instruments and experimental steps were involved. Rather than showing every detail, we selected 2 representative steps and illustrated them using AI-generated videos. For sample preparation and data collection, real experimental footage was incorporated and combined with experimental data to generate the final AI-based visualizations. Because the goal of this video is to explain the overall experimental design and research logic of the paper, the figures and supplementary videos from the original publication were used directly when presenting the results and conclusions. The following sections will walk through the production details of each segment. Most of the AI-generated clips in this video were created by providing either a starting frame or key frames at the beginning and end, and generating the motion using the open-source model WAN 2.2. This approach helps ensure that at least the opening part of each clip matches the intended composition and theme. The goal of the first segment was to visually illustrate how DNA shapes the resemblance between parents and their children. I generated a starting image using NanoBanana, depicting a family of 4 sitting on a sofa. The prompt only requested subtle motions, such as blinking and gentle smiles. However, in practice, the initial results showed movements that were too exaggerated, making the scene look unnatural, with frequent artifacts in the hands. By gradually reducing the described motion to an almost static level, these issues were largely avoided, but this also caused the animation to appear overly still, resembling a nearly static image rather than a natural video. The second segment illustrates signal transmission between neurons. I first generated several candidate starting frames using NanoBanana or SD 3.5, selected one that appeared reasonably consistent with known biological structures, and then used WAN 2.2 to generate the signal propagation animation. As neuroscience is not my primary field, even after careful parameter tuning, the flowing light used to represent signal transmission may still contain inaccuracies at a finer level. The third segment presents the hierarchical organization from chromosomes to nucleosomes and ultimately to DNA. I started with a schematic image from Wikipedia, applied artistic stylization using NanoBanana. The fourth segment was taken from the supplementary materials of a previous publication. This was followed by a presentation of the paper's front page, where the original figure was used and explanatory masks were added in DaVinci Resolve to illustrate the experimental design. No AI-generated video was used in this part. For the experimental workflow scenes, the shot of removing a flask from the shaker was created by first generating a starting frame and then using AI to generate the motion. In contrast, the step of adding solution into centrifuge tubes was generated directly from text prompts. In these laboratory scenes, limitations in training data often led to AI-generated results that violated basic experimental principles, such as failing to account for the need to symmetrically balance centrifuge tubes. For more complex sample preparation procedures, I therefore chose not to rely on AI-generated videos. When creating the segment that transitions from the interior of the electron microscope to the resulting sample data, NanoBanana Pro had just been released, and its generated starting frames appeared noticeably more refined than those from earlier models. The remaining segments were drawn from the supplementary videos of the paper. Animations that precisely track molecular motions are typically generated using specialized structural biology software, where atoms are interpolated from one defined state to another. When the two states differ substantially, intermediate states must be manually constructed. Because such datasets are scarce and highly process-specific, this type of visualization still largely requires manual generation in the near term. Overall, current image and video generation models are primarily trained on everyday scenes, and their understanding of domain-specific scenarios, especially the fine details involved in scientific research, remains limited. In practice, generating videos from a well-designed starting frame generally yields more reliable results than text-to-video alone. Even so, producing a satisfactory clip often requires repeated trials with different random seeds, combined with iterative prompt refinement. As a result, when visual rigor is critical or when specific experimental details must be accurately conveyed, manual recording, professional visualization software, or original figures from the publication are still the preferred choices at present. What follows is the full, uninterrupted version of the video.
DNA carries genetic information that, on a macroscopic scale, shapes the shared physical traits observed within families. At the microscopic level, DNA regulates cellular activity through gene expression. Chromatin remodeling and transcription are the initial steps through which DNA exerts its biological functions. Under the action of RNA polymerase, beginning at the transcription start site, nucleoside triphosphates pair sequentially with the DNA template strand and are joined to form RNA. This process is called transcription. In May 2025, our study published in Nature elucidated the early dynamic mechanism of human RNA polymerase III transitioning from transcription initiation to elongation. We designed a series of adenine-less sequences and added 3-deoxyadenosine triphosphate, allowing transcription to stall at defined positions for structural analysis of each stage. We recombinantly expressed and purified RNA polymerase III and its associated transcription factors, assembled them in vitro with U6 promoter template DNA to form transcription complexes, and initiated the reaction by adding nucleotides, including 3-deoxyadenosine triphosphate, to stall transcription at defined stages. The complexes were then applied to perforated EM grids and rapidly vitrified using a vitrobot, and extensive high-resolution cryo-electron microscopy datasets were collected to visualize the structural transitions from transcription initiation to elongation. Through a series of data processing and three-dimensional reconstruction steps, we obtained the structures of multiple complexes stalled at different positions along the transcription pathway. Focusing on the active center, the RNA-DNA hybrid formed between the nascent RNA and the DNA template reveals key pairing states during early elongation. From the global structural perspective, the relative positions of the transcription factors shift markedly, illustrating the dynamic rearrangements that occur during the transition from initiation to elongation. By using these complexes stalled at different transcription stages as key frames, we reconstructed the dynamic process of RNA polymerase III from transcription initiation to the synthesis of the first 13 nucleotides.